Hello, everyone, and welcome to today's live broadcast, CTS StemPro HSC Expansion Medium for Cell and Gene Therapies, presented by Abigail Becker, Scientist, Thermo Fisher Scientific. I'm Christy Jewell of Labroots, and I will be your moderator for today's event. This presentation is brought to you by Labroots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them in the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I would now like to introduce today's speaker, Abigail Becker, Scientist, Thermo Fisher Scientific. Abigail completed her master's degree in Biomedical Sciences at Penn State College of Medicine in Hershey, Pennsylvania, where her graduate research focused on the role of genetic and epigenetic alterations of acute myeloid leukemia. Abigail joined Thermo Fisher Scientific in the cell biology business based in Frederick, Maryland in 2016 as a R&D Scientist. Abigail is part of a team focused on research and development of cell therapy grade products that support translational and clinical research workflows. For a complete biography on Abigail Becker, please visit the Biography tab at the top of your screen. Abigail, you may now begin your presentation.
Thank you, Christy. Hello, I'm Abigail Becker, and I work at Thermo Fisher Scientific in the R&D department that focuses on cell and gene therapies. Therapies take life at the enablement stage, but there's a need to build in sustainability to develop these therapies into commercial products that can meet patient demand. Thermo Fisher has the global footprint and a history of providing high-quality technologies and regulatory support that clinicians need as they move to sustainable manufacturing of these new therapies. Our CTS products are developed to help ease the transition from the bench to the clinic. In addition to GMP manufacturing and quality systems, our CTS products provide traceability and regulatory support. Currently, our products are already in FDA-approved therapies and more than 100 clinical trials. One of our most recent CTS products focuses on hematopoietic stem cells, or HSCs. HSCs are self-renewing, multipotent progenitor cells that are found in the bone marrow and peripheral blood and are defined by CD34 surface marker expression. HSCs are of particular interest as potential treatments for blood disorders, immunological diseases, and cancer. HSCs can be genetically modified, for example by transfection or transduction as shown here on the right, and then transplanted into the patient. To provide long-term protection from disease, the modified HSCs must successfully engraft into the patient's bone marrow. In order for HSCs to engraft and repopulate the bone marrow, they must retain their full functionality, proliferation, and differentiation capacity. One of the major limitations to utilizing HSCs in cell therapy is the limits on cell numbers that can be harvested from a single bone marrow or cord blood donor. Expanding HSCs ex vivo would increase the number of patients that could be treated with a single harvest from either bone marrow or cord blood. However, ex vivo expansion of harvested HSCs results in rapid differentiation and progressive loss of CD34 expression. Current media systems support the CD34 positive progenitors that provide short-term bone marrow reconstitution. Long-term engraftment requires the true hematopoietic stem cells, which are thought to be CD34 positive, CD90 positive, and the CD45RA negative subset. However, this subpopulation of HSCs is currently not effectively expanded in commercially available media systems. To address this challenge, we used an iterative design of experiments approach to develop CTS StemPro HSC expansion medium with the goal of maintaining and expanding that rare CD34 positive, CD90 positive, CD45 RA negative population. The data on this slide focuses on CD34 positive cells from mobilized peripheral blood from 3 donors. The cells were expanded in CTS StemPro HSC expansion medium supplemented with stem cell factor, FLT3 ligand, thrombopoietin, interleukin-3, and interleukin-6. After 7 days, we evaluated the total nucleated cells, or TNC, and cell viability, and we determined the phenotype by flow cytometry using the Attune NXT flow cytometer. On the left, we show that StemPro HSC expansion medium provides about a 100-fold expansion in all 3 donors shown here, and that more than 60% of the expanded cells are CD34 positive. Note that while the competitor media expands both the total nucleated cells and CD34 positive cells, only StemPro HSC medium expands both the short-term CD34 positive progenitors, and it provides far superior expansion of the long-term CD34 positive, CD90 positive, CD45 RA negative cell population. Transplantation of ex vivo cultured CD34 positive cells is the gold standard to assess whether or not the expanded cells maintain their functionality. Here, CD34 positive cells from mobilized peripheral blood were expanded for 7 days in StemPro HSC expansion medium supplemented with the same 5 growth factors. The expanded cells were then transplanted into immunodeficient NRG mice. After 6 months, we monitored human CD45 positive cell populations in both the bone marrow and spleen. Obviously, you wouldn't expect any human cells in mice, so the presence of any human CD45 positive cells or chimerism indicates that these human cells have persisted at 6 months post-transplantation. This is indicative that the cells did successfully engraft into the bone marrow. When the cells that were cultured in StemPro HSC expansion medium were transplanted into the NRG mice, we see that human CD45 positive cells are 10-fold higher in bone marrow and 2-fold higher in spleen compared to the non-expanded cells. In addition, the cells exhibited multi-lineage engraftment, including CD34 positive HSCs, CD33 positive myeloid cells, and CD19 positive B cells. In addition to maintaining the CD34 positive stem cell population, StemPro HSC expansion medium supports megakaryocyte and erythrocyte progenitor cells. Megakaryocytes are the progenitors that differentiate into platelets, while erythroid progenitors differentiate into red blood cells. Here, CD34 positive cells from mobilized peripheral blood were expanded for 7 days in StemPro HSC expansion medium supplemented with the same 5 growth factors. At the end of the 7 days, we evaluated total nucleated cells and cell viability. In addition, we looked at the immunophenotype and determined the percent expression of CD34, CD41, and CD71 using the Attune NXT flow cytometer. Here you see a more than 2,000-fold increase of both the CD34 positive, CD41 positive megakaryocyte progenitor population, as well as the CD34 positive, CD71 positive erythroid progenitor population. Here we show CD34 positive cells isolated from the 3 most common tissue sources: 3 mobilized peripheral blood donors, 2 cord blood donors, and 2 bone marrow donors. The cells were expanded in StemPro HSC expansion medium, shown in blue, or one of 2 supplier media. Again, while all 3 media expand the total number of cells and maintain CD34 positive expression, as shown in the top and middle charts, StemPro HSC expansion medium expands both the CD34 positive progenitor cells and provides superior expansion of the long-term CD34 positive, CD90 positive, CD45 RA negative population. I'll move on to methods for genetically modifying HSCs for cell therapies. Here, mobilized peripheral blood CD34 positive cells were thawed and pre-stimulated in StemPro HSC expansion medium containing the same 5 growth factors for 48 hours. Cells were then transduced for another 48 hours with CTS LV-Max generated GFP virus at the various MOI as indicated. Transduction efficiency was evaluated by flow cytometry at 48, 72, and 120 hours post-transduction. On the left, the top row of dot plots shows the percent of cells that are double positive for GFP and CD34 at the MOI indicated. The bottom row of dot plots shows the cells that are triple positive for GFP, CD34, and CD90. Moving to the right, the middle 2 charts show the percentage of cells that express both GFP and CD34, as well as the mean fluorescence intensity of the GFP expression. In addition, all time points and all MOIs showed high viability. Together, this data shows that CD34 positive cells cultured in CTS StemPro HSC expansion medium can be transduced with lentiviral vectors with a 5 to 38% transduction efficiency when using the CTS LV-Max system. CRISPR-Cas9 gene editing allows genetic material to be added, removed, or altered at particular locations in the genome. Here, mobilized peripheral blood CD34 positive cells were cultured in StemPro HSC expansion medium prior to neon electroporation for the transfection of true-cut Cas9 protein, guide RNA, and GFP donor DNA. CD34 positive cells cultured in StemPro HSC medium showed approximately 20% transfection efficiency. And as a point of reference, T-cell transfection efficiency with this system is approximately 10%. Here we're showing the percentage of GFP positive cells, CD34 positive cells, and CD90 positive cells as determined using the Attune NXT flow cytometer. The center chart shows that CD34 positive, CD90 positive cells are present in the GFP positive population, demonstrating that these long-term HSCs are permissible to gene editing using this system. The far right panel shows the in vitro colony forming assay of GFP positive cells. Here we're showing that this resulted in granulocyte and macrophage colonies designated as white colonies here, as well as erythroid or red colonies. This indicates that the functional capacity of the cells has been maintained following electroporation. In summary, CTS StemPro HSC expansion medium was developed for cell therapy applications with regulatory compliance in mind. It is a serum-free, xeno-free, cGMP manufactured media system that is supported by a drug master file and regulatory support file to assist our customers in global regulatory filings and approvals. The media system expands CD34 positive cells from the most commonly used tissue sources and enriches for the rare CD34 positive, CD90 positive, CD45 RA negative subpopulation. It maintains HSC functionality and differentiation capacity and enables the expansion of CD34 positive, CD41 positive megakaryocyte progenitors and CD34 positive, CD71 positive erythroid progenitors. In addition, it facilitates engraftment in an animal transplant model and is compatible with gene editing and transduction workflows. Additional data and information can be found at thermofisher.com/StemProHSC. Thank you for your attention, and now I'd like to open it up for questions.
Thank you, Abigail, for your informative presentation. We will now start the live Q&A portion of our webinar. Also joining us today for our live Q&A, we have Jacqueline Ng, Regional Manager of Cell and Gene Therapy, APJ Region of Thermo Fisher Scientific. Now to our audience, if you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. Let's see, let's go with this question. And Jack, let's have you answer this one. Do I have to use the growth factors that Abigail has recommended?
Sure, I'll take this one. So for optimal performance, we recommend SCF, FLT3 ligand, TPO, IL-3, and IL-6. So that's the set of cytokines that we recommend. Using a design of experiments approach, we evaluated the impact of these growth factors individually, and in combination, we found the best performance when the 5 are used together. So the concentrations and catalog numbers for these growth factors can be found in our product manual online. But should you wish to add your own growth factors and use your own combinations, you could do so.
Thank you, Jacqueline. Abigail, let's come over to you for this question. Why is the CD34 positive, CD90 positive phenotype significant?
Sure. In 2017, there was a publication from the Fred Hutch Institute where they showed that the CD34 positive, CD90 positive population is highly enriched for stem cells, and that this subpopulation was responsible for multilineage engraftment in a non-human primate transplantation model. That paper was published in the Journal of Science Translational Medicine in 2017.
Thank you, Abigail. Now, the data showed 7 days of HSC culture. Will I get more cells if I culture for longer than 7 days?
Sure, I'll answer that one as well. So while greater numbers of cells can be achieved with longer culture periods, you have to remember that the percentage of CD34 positive cells is going to continue to decline over time in culture. So we evaluated culture periods that range from 3 to 14 days, and we found that 7 days is really optimal as it produces significant expansion of HSCs and maintains a high percentage of CD34 positive cells. So for example, at 4 days, we saw about a 25-fold increase in CD34 positive cells, and greater than 95% of those cells were CD34 positive, whereas after 7 days, we saw greater than 100-fold increase, and we were still showing more than 65% of those cells were CD34 positive. So we recommend 7 days. However, obviously, if people want to experiment and see what might work better for their needs, makes sense to do a time course study if you'd like to evaluate that further.
Great. Thank you, Abigail. Now, Jacqueline, let's come over to you for this one. What is the format of the media?
The media is sold as a kit containing a 50 times supplement, which is in a 10 ml vial, and a basal media, 500 ml basal media, that must be combined and supplemented with cytokines or growth factors before use. The supplement is stored at negative 20 degrees Celsius and can be stored in 4 degrees overnight or at room temperature for a few hours. Supplement should not be stored at 37 degrees. The basal media is shipped at ambient temperature and is stored at 4 degrees. So for more information, you can look through the FAQs on the product web page.
Thank you, Jacqueline. Now we're speaking of media. What type of GMP are CTS cell culture media manufactured according to?
I can take this question. CTS products are manufactured at sites that are ISO 13485 certified. They're compliant with US FDA Quality System Regulations 21 CFR 820. And they are registered with the FDA. While the products themselves are generally not medical devices, the manufacturing sites do manufacture medical devices, and the quality systems are aligned to medical device manufacturing standards.
Thank you. Now, Abigail, let's come over to you. In what system were the cells expanded? Bioreactor, flasks?
So we evaluated from like multi-well plates, I think down to 48 well plates, up to larger T flasks. We haven't completed any work yet in either like bioreactors or G-Rex, but at the moment, scale up through to large T flasks, we're showing equivalent expansion regardless of the 2D culture system, and we do hope to generate some application data for larger scale culture formats.
Thank you, Abigail. do you have any suggestions regarding most efficient methods of cell culturing, suspension, adherent, R&D phase, bioreactor?
So again, at the moment, we can really only recommend what I've looked into, which is the sort of smaller scale 2D culture. In terms of a bioreactor scale up, I would encourage anyone to reach out to us, and perhaps we could help come up with some initial testing for people to establish protocols. But at the moment, I don't have any recommendations for larger scale up.
Thank you. And Jacqueline, can we come to you for this question? How do I request a DMF or regulatory support file for this product?
You could go to our Thermo Fisher website and request a letter of authorization. You would need to complete an online form outlining the products that you are using, the clinical application reference number, title, agency division, and your company contact details. We will process your request and contact you for further information if required. So most requests can be completed within 1 week.
Thank you, Jacqueline. And looks like we'll wrap with this final question. In your DOE for the growth factors, did you also evaluate different concentrations for each factor? Or can you share what concentration that you used?
Yes, so I did a full DOE screen on the 5 growth factors we recommended. So I looked at least 3 concentrations for each, as well as, so I did this analysis or the data analysis, as well as the experimental planning using the JUMP software. So that allowed me to not only recommend an ideal concentration for each growth factor, but it also confirmed that all 5 growth factors really do provide a benefit in terms of cell expansion. The concentrations that we recommend are listed on the product page or the product insert. And again, as Jacqueline mentioned earlier, if anyone would like to evaluate additional growth factors, obviously they're welcome to do so. But we did really screen these growth factors and put a good bit of work into establishing what we think are the best concentrations across a variety of tissue sources as well as individual donors.
Thank you, Abigail. And thank you, Jacqueline, both for this interesting Q&A session. Do either of you have any final comments for our audience?
If you have any technical questions.
Sorry, go ahead.
Go ahead, Esther. No, I was going to say that if you've got any technical questions for us, you could write into our tech support email box or go online to our Thermo Fisher Scientific support website.
And I was going to suggest if anyone has would like additional information or would like to review any of the application data that we've shown here, you can go to thermofisher.com/StemPro HSC. There's a lot of great information as well as FAQs.
Thank you both for your time today and for your interesting presentation. Before we go, I do want to thank our audience for joining us today and for their questions. Those questions that we were not able to answer today or those that are submitted during our on-demand period, they will be addressed by our speakers via the email address that you provided at the time of registration. I'd like to once again thank Abigail Harris-Becker and Jacqueline for their time today, and we would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and Labroots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon. Have a great day.